Okay, it's all you. Go ahead.
Okay. Well, real quick, it's important for me personally to acknowledge that the research and the learning that I take part in mostly happens on Squaxin Island tribe lands. And these lands are so amazing and beautiful, and I get to appreciate them because they've been stewarded for, since time immemorial by these tribal members. And I want to, show my appreciation and thanks for that. And thank you all for being here today to hear me discuss my research. My thesis is on the seed production and viability of a native plant, the golden paintbrush, Castanea levisecta, uh, comparing native and reintroduced populations. That is wild legacy populations that have been here also since time immemorial, and the populations that restoration science has tried to bring back through outplanting and seeding. I'm going to give you a quick introduction just to the situation with golden paintbrush and my connection to it. This is a very rare native plant species. There are only a few dozen sites of relatively modest size, only several dozen to several hundred acres where it persists, and it's been the focus of a lot of restoration effort and work and time and money. And its story started in the 90s when people started to notice that it was disappearing. Sites were being developed. Its habitat was being destroyed. Populations were no longer as prevalent as they used to be. And it was listed in 97 as a federally threatened plant. Um, at that point, work began, and you can see that on the population graphs in the middle of my screen here, at the very top, you can see a skyrocketing outplanted population, that orange line at the very top of the screen. And that was really great news for the plant as a whole, and led to it being delisted just very recently in 2003, as the overall population is stable and abundant to a point. Unfortunately, that doesn't tell the whole story. I work for the DNR, as Sarah told you earlier, and one of my tasks is monitoring several populations of Castanea levisecta, and I have seen year by year that the native populations have done very poorly, as is illustrated by the blue lines in all three graphs there. The bottom graph depicts my original study site, Rocky Prairie, where the populations have done particularly terribly. And I'm wondering to myself as I go about my work, is a genetically isolated population, and these native populations are in fact genetically isolated, as opposed to the outplanted restoration populations, which are mixed. Are these populations getting what they need? And specifically, are they producing fewer and less viable seeds to replenish themselves? A little background on the plant itself real quick is golden paintbrush needs a minimum of 1,000 individuals in a population to be able to sustain itself, and there needs to be genetic heterogeneity within those population individuals. It does extremely poorly when self-pollinating or pollinating with a genetically related individual plant. The habitat quality is a big deal. Low stature vegetation prairies that have the plants that golden paintbrush can live alongside of. It's a hemiparasitic plant, which means it bonds with other plants in its environment to grow more vigorously and reproduce. Without that type of environment, which is very, very, very rare now, the Washington prairies are at 2 or 3% of their historical coverage. Without those things, it's having a very, very hard time. Pollinator support is another big deal. It's not a wind-pollinated plant. It needs primarily bombus species, bumblebees, to pollinate it, and many of its sites are too far away for those pollinating insects to, go from one to the other. And there are things like highways and developments in the way to keep them from doing so. But management activities, have really gone wild over the last 30 years. Preserves have been established. Propagation and reintroduction has been happening all over the place. There are multiple nurseries that grow this species for restoration. And there's just been a huge push in bringing it back. But there are still quite a few threats that challenge this restoration effort. One of the largest ones is inbreeding. With plants not able to genetically mix with each other as their sites are far away and their native populations are being genetically isolated. In the literature, it shows that this leads to reduced seed set, and it's a big problem. Invasive species obviously are a problem for most native plants, and this is no exception. Habitat loss, as I had talked about earlier, with development and such like that. Pollinator loss, those bumblebees not showing up at the right time of year or frequently enough to propagate these populations. Herbivory is a minor issue, but it really goes along with habitat loss, as the majority of the herbivory on these plants would come from grazing cattle. And hybridization is a sneaky little problem as well. I've included some images here because they're just so beautiful. These are images I've taken in the field of these plants producing hybrids with other Castanea or paintbrush species, and the seed produced by those species are not viable for restoration work and dilute seed resources when they contaminate our seed products. So it's a bit of an uphill battle still. The methods for my specific study, which are examining the seed production in count and seed viability and germination rate. Additionally, the weight of Castanea seed is detailed here. And my study site, Rocky Prairie, and the counterpart site, Mima Mounds, represent a native population and a restoration population, Rocky being the native population and Mima being the restoration population. They are roughly 14 kilometers apart. You can see that little red line near the bottom of the screen. It points to both of the sites. And a sampling plan based on 100 plants was developed. I was able to get and sample 100 seed pods, one seed pod per individual plant at Mima Mounds Prairie, with a distribution of samples that was equal to the distribution of plants in the different restoration segments, which you can see illustrated on the upper map there. Unfortunately, Rocky Prairie's population is doing so incredibly poorly that there were only 48 plants that were able to produce seed pods for me to sample. And to put that in perspective, about 13, 14 years ago, there were over 5,000 plants on the site, maybe around 6,000. And just a few years ago, there were 1,000 plants. So it's a very, very present and rapidly worsening problem for this, which is the most populous and healthy of the local Washington native Castanea sites. This is the best we have to offer. So it's really, really bad. Anyway, these numbers I pulled out allowed me to capture a 0.04 effect size at a 0.8 or 0.4 effect size at a 0.8 power at a 95% confidence interval, which will allow me to show some serious differences if they're there. My methods, once I got the seeds in my hands, were I kept them stored for a while until I had worked out my methods. And then I counted individual seeds, these tiny, tiny little seeds. They're about 0.2 of a millimeter long. I counted well over 10,000 of them, and then weighed each seed pod's group of seeds to get an average weight per seed. And that was the lab work that I did, and the seed quality work was done by the Oregon State University Seed Lab. I sent the seeds from either site to receive tetrazolium seed viability testing, which told me what percentage of viable seeds were present in each sample. And then the germination rate test would tell me actually how many seeds from those samples germinated. Those were two separate tests. And all the analysis of statistics was done in RStudio. The results of that seed count data are depicted here. The box plot in the far upper right corner shows the descriptive stats below it. But it details here that Mima Mounds, the restoration site, the non-genetically isolated site, is producing much, much more seed per pod, and therefore per plant, than the native legacy site, Rocky Prairie. Like, it's not even close. Almost 3 times or over 3 times as much seed, which is a huge, huge problem because, well, we'll get into seed viability later. But so we know one of the reasons this might be happening is the inbreeding, but I personally didn't think it was going to be this stark of a difference. The Rocky is producing lower seed at the top, or less seed at the high end of its range as well. Mine was well into the 200s, and Rocky's barely over 150. So that's a big problem. And the histograms you see in the middle of the screen, they visualize the difference in distribution in amount of seed produced per pod. And you can see that huge, huge column of zeros for Rocky. Most of Rocky's seed pods that I collected had, well, not most, but many, over a third had no seeds in them at all. It's by far and away the highest frequency. And that's just really, really bad. So moving forward here, I want to get into the viability results. Now, due to the extremely small amount of seed available for these plants, only one test was able to be run in the lab for each of these methods, the tetrazolium viability and the germination. So anything to be gleaned from the results is very superficial and really just possibly pointing the next direction to go with study. But they're listed here for viability. Rocky Prairie actually had a higher viability rate of the seed produced, which surprised me, but definitely I think alludes to a strong genetic legacy at this site where, you know, just a few years ago, there were thousands of plants. So that bodes well for recovery. High viability is good if we can get them to crossbreed and produce more seed. Perhaps we can get more plants out there. Mima was only viable at a rate of 1/3, which with so many more seeds produced at the site might not be as big a deal. But as far as the germination result, those turned out to be pretty fishy. A germination result of 2% and 0% makes me really wish that I had been able to run more samples, because that depicts an error on my part, or the part of the lab, but probably my part with the stratification process. It just seems like a wetter stratification might have yielded more, as is alluded to in the research, but I got mixed messages from the research. That's something that we'll be exploring in the future, hopefully, if I get a chance. The seed weight results are, these seeds returned results that showed Rocky was quite a bit heavier than Mima on average, but these are to be taken with a grain of salt, as there's huge, huge variation in the standard deviation of the measurements taken. I used a calibrated analytical balance to do this, which goes down to I think, a thousandth of a gram or maybe a 10,000th of a gram. And there was still massive fluctuation, and some of the numbers even ranged into the negatives, which is definitely impossible. So while I trust the implications given by the average of all these readings, anything beyond that is a little suspect. Moving forward here. Okay, so the meat of this, the seed count. It's clear that wild populations, this population in particular, are producing less seed than restoration populations. And that's basically the exact thing that I expected to see from the literature. These populations have been genetically isolated by management decision, these wild populations, for many years, and that can't not lead to self-breeding in the population. And this species, it is shown in the literature, suffers pretty severe inbreeding depression problems. And I think, in my opinion, that's what's happening here. Both of these sites produced lower than what the literature says their average seed production should be. Those numbers from the literature are several years old. So I wonder if that alludes to other problems, possibly with pollinator populations or unstable weather conditions that are influencing seed production as well, outside of inbreeding depression. But with Rocky Prairie producing less than a fifth of the seed it's expected to, something has to be done right away. Especially with only 48 plants producing seed last year, out of what should be thousands. The restoration population is doing pretty good. They're producing, 318%. I calculated what the wild population is, but that doesn't really mean much when a wild population is doing pretty poorly. As I said, they're still lower than the average seed production from the literature. But the differences in distribution that were detailed on the earlier slides, I think, may have something to do with this. I'm not sure exactly how it would be worth investigating them further, but it can't bode well to have so many empty seed pods. Moving forward, discussing the viability and germination rates. With the restoration site being, this single test implying with the restoration site being about 70% as viable as the native legacy site, I think it's important for us to get more of that legacy seed out onto the rest of the prairies and start crossbreeding it, both for the purposes of supporting those wild populations with new genetics and supporting these restoration populations with hopefully more powerfully viable seed. But really, more samples need to be run through that testing procedure, and maybe we can make some management decisions based off of that in the future. As far as germination testing goes, as far as germination testing goes, I really can't draw any too serious conclusions off of a single sample run that I think may have suffered from a little user error. But I am going to be attempting to run, if I can get enough seed, run this test again, possibly with replication, to test the true germination rate of these two different types of populations. And so for conclusions and recommendations, I've gone over most of them already, but the wild populations, specifically the wild population I studied, and by implication, other wild populations that are doing similarly poorly, they're just producing less seed. And that reduced seed level, I can only determine that it is driving, or at least partially driving, the population decline at those sites. We know that genetic isolation contributes to inbreeding depression, which contributes to reduced seed set. So the isolation at these sites must, or is very likely, contributing to the reduced seed production at said sites. Inbreeding depression is something we have to address in these wild populations as soon as possible, like right away this coming season, if we can, with such drastic drops in population. And now looking at double, and at some sites single digit population numbers. I don't think we can afford to wait very much longer. But it's clear that our management decisions and practices should no longer include isolating those populations genetically. Anyway, thank you all for coming, and I hope my voice came across clearly. If you have any questions, I would love to answer them now. I think I have quite a bit of extra time.